All right, hello everyone. It's an honor to be introducing our next speaker in the NHGRI seminar series. This talk will be recorded, so if you're interested in catching it or sharing it with others, it'll be available online in about a week. Second, if you're attending online, welcome. You can put in questions into the Zoom Q&A at any point, and then at the end of the seminar, someone in the room will relay your questions to Lars. And then third, if you're in the room, also welcome. There's a microphone, so at the end of the talk, feel free to queue up to ask your questions, or we can also walk around with the microphone if you raise your hand. Okay, so our speaker today is Dr. Lars Steinmetz of genetics at Stanford University and also a group leader at Heidelberg, EMBL Heidelberg in Germany. And so that's right, Lars runs two groups in two different countries. And I think this is really like a physical manifestation of the breadth of Lars's research, where Lars does research spanning from basic genetics research all the way to clinical genetics research, just like he is literally spanning the globe himself physically. So these twin themes of basic research and clinical research have been present in Lars's research from the very start. When he was a graduate student at Stanford and as a postdoc, he did work in basic research studying quantitative traits in yeast and doing fine mapping of specific genes involved in high temperature growth, really helping to understand really the genetic basis of complex traits that involve many genes across the genome. At the same time as he was doing this basic research, Lars was also working to understand mitochondrial disease genetics to identify potential disease genes underlying mitochondrial disease. And these two themes have continued in Lars's work up until the present day. So today, Lars will be telling us about his work on dilated cardiomyopathy. And as you can tell from the title, he'll be telling us, even for this one disease, spanning all the way from understanding the disease, diagnosing the disease, and all the way potentially to curing and treating the disease. And towards this goal, Lars really brings to bear really exciting genome technologies and other technologies. So I don't know if he'll be telling us about this today, but he does work with like CRISPR screens, base editing as a gene therapy, and a new technology that to me seems basically like science fiction, which is image-based FACS analysis. So with that, Lars, we're very excited you're here and excited about your talk.
Thank you for the kind introduction, Meru. That was a nice introduction in terms of topics that I'll cover in my talk today. As Meru said, my lab is interested in understanding how genetic variants lead to phenotype, and we've been primarily interested in a disease application, dilated cardiomyopathy, over the last few years. And we really took out to take all the tools we have available to us from the basic sciences and apply that to this problem to come up with new therapeutic interventions for patients that have genetic forms of this disease. And so this is really inspired by the fact that we're seeing a transformation in the medical system that's informed by genetics that enables us both to target diseases in a more mechanistic and targeted fashion, to both cure them when they've onset, or even to think about preventing the diseases in individuals that are carriers of mutations, for example. I've depicted it like this because I actually think the preventative strategy ultimately will be an easier route to go from a patient perspective than trying to fix a disease when it's already manifested itself and progressed. And in the particular case of cardiomyopathy, where the heart has already malformed, dilated in the case of dilated cardiomyopathy, and it's pretty much impossible to go and reverse course again. If we could understand the molecular mechanisms and detect them early, we might be able to intervene and prevent these diseases from arising in the first place. So my lab started to approach this problem from the angle of genome sequencing. We had been sequencing individuals, in particular here, a healthy individual that we did whole exome sequencing on, and as now there are over 100,000 variants that have been implicated with phenotypes, we can simply look up the variants that are predisposing to disease in any healthy individual's genome. And this is an example here. I've just highlighted on the right a couple of disease genes and diseases that show up. You see a lot of variants that predispose to cancer in this particular individual. But you also see a famous, or the famous ApoE4 variant that James Watson didn't want to know about when his genome was sequenced because it predisposes to Alzheimer's disease. But ApoE variants, in particular heterozygous variants, are not really rare. 24% of the population is a carrier for such a variant. So I think in this case, not something really to worry about. The only real variant this individual needs to worry about is an RBM20 variant that predisposes to dilated cardiomyopathy. And this particular variant is a dominant mutation. It explains, or RBM20 mutations explain about 3% of dilated cardiomyopathy cases. The penetrance is very high, on the order of 80, 90%, and high mortality rates with an average life expectancy of 45 years in some families that have mutations within the same region of this particular gene. So the obvious question comes when an individual that doesn't have disease has a variant like this, what can I do about it? And if you actually look at the current catalog of genes that are annotated as being actionable in the human genome, the American College of Medical Genetics and Genomics currently classifies 95 genes as being actionable. So these are variants where it would be important to know whether you're a carrier with a disease mutation in those genes, as you can do something about it. But considering there are hundreds of thousands of variants currently annotated through GWAS studies as being potentially disease-causing, we would like to expand that list. And so more knowledge needs to be generated to understand what these genes do and to come up with mechanistic recommendations to prevent these diseases. So as Mero mentioned, my lab is really interested in genomic technologies broadly applied to reading, editing, and writing genomes. And we apply these in various thematic areas. One of the areas is in single-cell functional genomics, where we carry out genetic CRISPR screens using single-cell readouts to understand biological function. We have projects that focus on precision genome editing to engineer using CRISPR variants into genomes and as well as into model systems for disease to study them. Some effort in wearable technologies and devices in order with the idea to monitor health status, more routinely detect early onset of diseases, and then areas where we take all of these tools and apply them in a disease context. And one of these areas is dilated cardiomyopathy. So in my talk, I'll touch on some of these aspects that are all coming together in the context of dilated cardiomyopathy research as we've applied it. Cardiovascular disease is one of the main causes of death. In fact, it explains about a third, roughly, of deaths worldwide that occur. It manifests itself in various forms, usually with the ultimate outcome being heart failure that occurs, but there can be subclassifications of heart valve disease, for example, areas where the heart muscle is affected. These are cardiomyopathies. These cardiomyopathies can also coincide with arrhythmia that occurs where the heart doesn't beat anymore in its normal rhythm. And then there are vascular diseases like coronary artery disease where blood flow is being impacted and also will impair the both the heart's supply of oxygen to the muscle cells and also the distribution of oxygenated blood throughout the body. Currently, diagnosis for heart diseases are carried out using primarily echocardiograms, which can measure the ejection fraction of the heart and give you an idea of the pump function of the heart, or electrocardiograms that can measure electrical transduction through the heart, and MRIs, which are carried out that are higher resolution than the echocardiograms and can show you malformations or enlargement of the heart. Usually blood tests are also carried out for biomarkers. Troponin T tests and BNP, for example, are two proteins that are secreted by heart muscle cells when the heart suffers into the bloodstream and can be detected. And usually genetic testing is recommended when an individual comes in where there might be a family history of this disease to try to identify one of the causal genes that are currently known. In terms of treatment recommendations, there are medical interventions available. Usually these medicines reduce the load on the heart. So for example, angiotensin converting enzyme ACE inhibitors or ARB blockers or beta blockers are used. So simply reduce the workload that the heart experiences. Devices can be implanted, in particular pacemakers, for example, on individuals that experience arrhythmia, and usually lifestyle recommendations in terms of diet and exercise are recommended. And for advanced cases of heart failure, unfortunately, the only real treatment option or cure that exists to date is a heart transplant. And for many patients, that is not an option if they don't meet certain age requirements, et cetera. So we really would like early detection of these diseases and more mechanistic and targeted therapies to be developed. So we are particularly interested in dilated cardiomyopathy, which is a form of cardiomyopathy where the left ventricle expands and enlarges in size, so the heart overall gains more volume, and ultimately the muscle walls become so thin that they're unable to pump the blood through the body, and these individuals then experience heart failure. It's characterized by a reduced ejection fraction and has a prevalence of about 1 in 250 individuals. So isn't entirely rare. About 30 to 50% of the cases of dilated cardiomyopathy are due to a genetic defect and are familiar, so they'll segregate in families. And to date, over 20 genes have been identified with mutations that can lead to dilated cardiomyopathy. Of these, we're particularly interested in RBM20. RBM20 is a key splicing regulator of the heart, and it actually also controls the splicing of some of the other genes on this list that themselves, when mutated, can lead to dilated cardiomyopathy. We're interested in this because it also leads to a particularly aggressive form of dilated cardiomyopathy. Most of the mutations, as I mentioned, are dominant, so a single mutation is sufficient to trigger the disease. It's also associated with malignant arrhythmias and high rates of mortality, including sudden cardiac death, probably due to the arrhythmia that's triggered by the disease. The main mechanism of function when we started to work on this disease was to think about this as a splicing disorder. Since RBM20 is a key cardiac splice regulator, you can see the protein diagram sketch on the top in terms of different domains that it contains and annotated mutational positions. And you can see right away that there's a hotspot region of mutations right around the RS domain, the arginine serine-rich domain, where there are actually 5 amino acids in a row that all have been previously associated with dilated cardiomyopathy in independent families. And there are also some additional mutations sprinkled outside that. So for example, in an glutamate-rich region or in the RNA recognition motif region where individual mutations have been identified, the vast majority, though, are within this arginine serine-rich region. RBM20 is known to control the splicing of key cardiac transcripts, titin being probably the most famous among these. That's the largest protein in the human genome. And it actually also determines the stiffness of the sarcomere, and so can control stiffness of the heart, in particular during contraction. And when missplicing and titin occurs, then you generate a more flacile form of that protein that probably is less rigid and therefore can contribute to the dilation that's observed in the heart. We got to this, as I mentioned already earlier, by sequencing a family in which dilated cardiomyopathy was segregating and ultimately could then link a new mutation within the RS domain to the disease in this particular family. The mutation that we identified is called P633L, shown here in red. It's one amino acid adjacent to this series of 5 amino acids in a row that had been previously identified. So it really suggested that this mutation would cause the disease in this family too. And of course, we've validated that in cell-based systems and also could show that this mutation leads to missplicing of known RBM20 target genes, titin being among them, but also genes that control electrical activity of the heart and ion homeostasis like the ryanodin receptor 2, and also signal transduction molecules like the CAM-K2D molecule. So it affects both the structural structure of the heart, but also the timing of contraction that occurs, and thereby could explain both the dilation and the arrhythmia that one can see in the system. We then set out to generate models in the lab for how we can tackle that and approach this further and gain better mechanistic insights in how this mutation really leads to the disease, and then ultimately how we can intervene. So for that purpose, we set up model systems. One of them was iPS-derived cardiomyocytes. This enables us from blood cells to generate iPS cells from patients and then differentiate those into beating cardiomyocytes that one can analyze in the dish. And then we can apply our genetic tools, CRISPR agents, in order to perturb these and study them in more detail. We also wanted in vivo models, because ultimately the heart functions in an organism context, and we want to be able to measure pump function and implications of that. So we needed animal models. We set out to generate mice models in which we used CRISPR to engineer in the exact patient mutations into the orthologous positions of the mouse. RBM20 is highly conserved. There's only a 2 amino acid shift in the annotation, but otherwise all the amino acids within the RS domain are exactly identical. So we made our new mutation as well as a known mutation right next to it that was previously characterized. And we were fortunate enough that a company who was working on a pig model contacted us to collaborate and characterize their system in more detail. And that was Jay Schneider and Recombinetics, who had used TALENs to generate an RBM20 RS domain mutation into the pigs using TALENs. And we started to characterize these in detail. In terms of tools that we could apply to the problem, then we used first transcriptional characterization with long read sequencing to actually define all the splice isoforms that are being affected by the RBM20 mutations. With this technology, you can sequence full-length cDNA by simply pulling the cDNA through the poly-A tail through one of these nanopores, and therefore sequence the RNA from the poly-A tail all the way to the 5' end, and get complete information on splicing that has occurred across target transcripts of the genome. And this we applied to iPS-derived cardiomyocyte RNA messages that we converted to cDNA and then full-length sequenced them genome-wide, and then analyzed these data between wild-type iPS cells and then our 2 mutant models, the novel mutation P633L and then the previously known R634Q mutant that had been characterized before. And what you can see here as an example is the long read data. I'm showing it here for one gene, IMMT, which is also known as mitophilin. It's a part of the micose complex that is involved in crystal formation in mitochondria. What you can see in wild type is that there are 5 different transcript isoforms for this particular gene that are expressed in cardiomyocytes. Each blue line is one long read that was generated. You can see these are there at slightly different abundance, but all are easily detectable. In the mutants, you see an absence, in particular, 2 isoforms that are completely gone here in this mutation and reduced drastically in the more subtle P633L mutant that we characterize. But essentially, so very specific alterations on splicing are observed due to these mutations. And since we had genome-wide data, we can simply ask genome-wide, what are all the transcript isoforms that are altered in the mutant versus the wild type? And so this type of analysis allows us directly to do a differential expression test at the level of isoforms rather than at the level of genes, and directly pull out the isoforms that are affected. And we thereby associate about 100 transcripts, 100 genes as being misspliced due to the RBM20 mutations, highly enriched in cardiac function. So these give us an idea. Then you could further mine that to look at potential targets and try to associate those with mechanism. What had happened in parallel to us working on this is that we got access also to the pig material from the RBM20 RS domain pigs that were generated. And Jay Schneider characterized the localization of the RBM20 protein. And what really became apparent here from these data is that the disease has much more to it than a splicing disorder in the nucleus. What you also see in the mutant pigs, and the same is true in mice and in humans, is that the RBM20 mislocalizes. It's supposed to go to the nucleus where splicing occurs, but in the mutant cases, you get accumulation in ribonuclear protein granules in the cytoplasm. You can see that very nicely in the wild type. If you look at RBM20 labeled in red, it is located in the nuclear compartment, and particularly in splicing speckles. These are factories where splicing occurs, particularly driven by titin splicing, where also other target genes kind of come together in a 3D conformation at the nuclear level to coordinate splicing of RBM20 target genes. But if you look at then the mutants, both the heterozygous and in particular, if you look at the homozygous case, you don't see the splicing speckles anymore in the nucleus. You see red dots in the cytoplasm, which are these ribonuclear protein granules. These are liquid phase-separated entities that still are not very well characterized, resembling potentially Peabody-like structures that also seem to be contributing to the disease. And what was really key, I think, an experiment generated by Hideito's lab in Japan was this experiment where he characterized the mice models and compared the knockouts, the knockout RBM20 model to an homozygous point mutant RS domain mouse. And he could really show that the knockout actually doesn't have a survival decline compared to the wild type, while the point mutation homozygous animal has a survival decline and has a much more severe phenotype than the knockout. In both of those animals, you miss splicing due to RBM20 target genes. Since, when you have a knockout, you don't have RBM20 around, but you also don't have granules. While in the case of the RS domain, you have both missplicing and granules there. This suggests that the granules are also contributing to the disease phenotype and seem to have a dominant negative effect. And that resembles roughly to the clinical severity seen also in patients. So we then wanted to come up with therapeutic strategies based on these insights of how we might be able to treat this disease. And we could come up with 3 strategies that we then pursued in more detail. The first one actually, before we even knew about the granules, was to think about this as a haploinsufficiency on splicing. You have half the dosage of RBM20 in a patient. Can we increase RBM20 expression levels so that the wild type allele, which every individual carries, will compensate for the loss of function of the misspliced version, and thereby increase splicing on RBM20 target genes? And so we actually bioinformatically mined through databases to look for small molecules that were upregulating RBM20 expression in any cell system. We identified that all trans-retinoic acid in a publication had upregulated RBM20 in an embryonic stem cell system. So we took all trans-retinoic acid, which actually is vitamin A, applied it to our iPS-derived cardiomyocyte system, and could also show that in that system, RBM20 levels increase. And we could actually show a roughly doubling of RBM20 expression level that also restored then the splicing on RBM20 target genes. So this seems to be a viable strategy, in particular in cases where you have missplicing as the main driver of the disease for RBM20. But we were worried about the granules. So if you upregulate RBM20 expression, you presumably also make more cytoplasmic granules, which could have a dominant negative effect ultimately in an in vivo system for RS domain containing proteins. So we came up with 2 other strategies to tackle this. One of them is focused on restoring localization of RBM20, which is not getting to the right place in the system, with the focus that if we could bring mutant RBM20 protein back into the nucleus, then potentially it would still function in splicing and we could restore splice activity. And a third strategy, which is focused on gene editing, would be to directly fix the alleles in the heart in vivo by targeting an editor to cardiomyocytes and then fixing the genes in the system so that you would never develop the disease in principle if you even would apply this early on. So we pursued both of the strategies on the right in more detail, and I'll actually tell you where we are to date on both of those 2 investigations, starting with the idea to restore RBM20 localization back to the nucleus. And to tackle this problem, we actually used the technology we had developed in the lab for an entirely different purpose that we could then apply to that Mero mentioned initially, image-enabled cell sorting. So this came out of a collaboration with Becton Dickinson Biosciences in San Jose, who are the leading developers of FACS instrumentation, and they had invested about 10 years of development into generating a device, a FACS machine that combines microscopy and FACS in one system and would actually enable one to image cells on the fly as they pass through the FACS sorter, and then use image analysis in order to make sorting decisions, so that ultimately you could sort cells based on their subcellular localization of a protein tag, for example, instead of relying on surface markers. And so that opens up the space of kind of genetic screens significantly to much more rich phenotypes that we can collect based on some microscopy-like data sets. The way the system works, as I mentioned, so it's just built around a standard FACS machine. You have cells passing through a laser beam, and then a sorting decision is being made. What makes image collection on the system possible are two innovations. The first one is the ability to extract images at sorting speeds. And that uses a technology called FIRE, which Eric Diebold, who is at BD now, also developed and published previously on. This approach shifts the frequency, the radio frequency slightly of the laser beam, and dissects that down to about 100 different laser spots that are with a different frequency depending on the x-axis coordinate. So if you have an intensity associated with a frequency, you know exactly along which x-axis coordinate that data set was collected. And then the y-axis coordinate is simply the time or the velocity as the cell passes through the beam. So through that data, you get then two-dimensional image data of cells as they're being analyzed. And what you need is also rapid compute with low latency electronics in order to reconstruct these images on the fly and make a sorting decision within about 400 microseconds, which is the time frame you have when you sort at speeds of about 15,000 cells per second, which this operates at. So the way it works in practice, cell passes through the laser beam, an image is collected, image analysis is carried out, and then a sorting decision is being made. So by the time you reach the splitter, you can decide where to sort it into. For example, nuclear versus cytoplasmic localization of a GFP tag protein is accessible. The resolution of these data are pretty good. Here's just an example of what you can see. With such a system, you can clearly distinguish stains of the nucleus, for example, which you can see here from cytoplasmic localization, but you can also see smaller entities like P-bodies or centrosomes that can be visualized. And these are all images collected at these sorting speeds of several thousand per second. So this was obviously perfect in an application applied to RBM20 biology, because RBM20, as I mentioned before, is mislocalizing in the disease case and falling into the cytoplasm, while in the wild type, it should be in the nucleus. So we can then use this to set up a genetic screen to look for factors that are involved in localizing RBM20 to the right location. So we do this experiment by, again, GFP labeling RBM20 here in green, and then using DRAG5, which is a nuclear dye, to stain the nucleus. And we can then analyze these ICS-derived images. If you get high correlation between these two dyes, it would be RBM20 is nuclear localized. A low correlation indicates cytoplasmic localization. And just to remind you again, we can clearly distinguish these phenotypes by microscopy, but also by image-enabled cell sorting then. You can see here, again, I showed it to you earlier, the splicing speckles in the nucleus and wild type, and then these cytoplasmic granules in the mutant examples. And microscopy also shows that there is a different degree of mislocalization depending on the mutation that you have. So in the P633L mutant, which is the more mild mutation also on our splicing analysis, we see fewer mislocalized proteins and more severe mislocalization, the R6Q mutant. So that coincides with the splicing phenotype we saw before, but also with the severity in the families that are segregating for this disease. And ICS is able to resolve that localization difference with P633L being in the middle. So having the resolution to really distinguish then these mutations from each other and also from wild type, we could then take this forward in a genetic screen context where we would use our whole genome library of CRISPR perturbations, perturbing every protein coding gene in the human genome, and apply that in the context of an RBM20 localization screen to figure out the factors and ultimately the transporter that's involved in bringing RBM20 from the cytoplasm into the nucleus. So this is done with using a lentiviral library, which contains and expresses a guide RNA. So each lentivirus will perturb one protein coding gene. We transfect that into a HeLa cell expressing RBM20 GFP, and then we sort that population, collecting the nuclear localized fraction and then the cytoplasmic RBM20 localized fraction, and then simply sequence these pools to determine which guide RNAs are present in the cytoplasmic versus the nuclear fraction, and that gave us a very clean hit. TMP03, you can see here as being really the major disruptor of nuclear localization. So when this gene is being knocked out, we no longer see RBM20 going to the nucleus, and it stays in the cytoplasm. And TMP03 actually has been described previously as a transporter for arginine serine-rich domain proteins, but never been associated with RBM20 before. So it made sense that this could act as a transporter. And we then validated that hit both computationally with AlphaFold predictions, where we model TNP03, the transporter, together with the RS domain of RBM20. And you can see that here, RS domain shown in red and in blue is mapping into the binding pocket of TNP03. And the predictions also suggest that the mutations disrupt the binding interface, with a more severe disruption in the more severe R644Q mutant and a more subtle disruption in our P633L mutation. And we validated that on biochemically as well with co-immunoprecipitation experiments, where we pulled down RBM20 and then asked how much TMP03 co-precipitates. In a wild type, you get a good pull down signal, while in the mutant cases, you're pulling down less. And again, the degree of severity of this interaction recapitulates at the biochemical level. So the key question then was, now that we know that the mutations disrupt binding to the transporter, would the mutation still be splicing active if we were able to get it into the nucleus? And would that then be an active way to tackle this disease? So to do this experiment, we took the mutant RBM20 protein and we fused it to a nuclear localization sequence. And the nuclear localization sequence would force the mutant protein into the nuclear compartment, and we could then analyze splicing. First, to show you the NLS fusion worked. So here in an R644Q mutant, again, cytoplasmic localization of the mutant. In the NLS fusion, it's going into the nucleus. And so RBM20 is being relocalized, and nicely for us to see was that splicing was completely restored when the mutant protein was brought into the right compartment. You can see all the splice changes in the mutant relative to wild type, but in the NLS fusion, we get active restoration of splice activity. So the model is then that TMP03 and RBM20 interact through physical binding. TMP03 brings RBM20 into the nucleus, where it can actually splice its target genes in the mutants within the RS domain, which is the interacting interface with TMP03. You get disruption of this binding, so less RBM20 makes it to the nucleus, and so you get a splicing deficiency on RBM20 target genes. And the idea here would be that we could then perhaps exploit this by increasing RBM20 localization by, for example, overexpressing the transporter and supplying more transporters so that mutant RBM20 would be brought back into the nucleus to more extents. And so we tested this latter approach simply by packaging TMP03 in a viral vector, an AV9 vector, that would then deliver TMP03 to heart cells as well as other cells in the body. And the question here was, in an animal model now, when we go to mice, could we restore RBM20 phenotypes in this animal model in which mutations in the RBM-RS domain are present? So we tested mice with both the P633L mutation and the R644Q mutation. Again, there's a 2 amino acid shift in the annotation, so that's why the numbers are different between mouse and human, but these are orthologous positions. We take mice at 4 weeks old, which are essentially juvenile mice that are being injected, and we analyze them at 16 weeks, which is then an adult mouse. And we did localization analysis. We can show also in the mouse, like in the cell system I showed you earlier, that TMP03 overexpression brings RBM20, mutant RBM20 back into the nucleus, also in the in vivo context. We see as a restoration of splice activity also in vivo, like we had seen in the cell-based system, where you see defects in the control samples and restoration towards wild-type levels upon TMP03 overexpression. And most importantly for us is that the pump function of the heart was restored too when the splicing was corrected. So you can see a significant ejection fraction decline in these mutant animals, both for P633L and R634Q, and that ejection fraction is restored to near wild-type levels when TMP03 is overexpressed. So that provides proof of concept that we could target these diseases by focusing on trying to get mutant RBM20 back into the right location. So this is a project that's further being worked on. We're now thinking about other ways in which we can do the relocalization besides overexpressing the transporter in more specific and targeted ways. And that's one of the areas that are still being pursued. I want to tell you about this third approach that we tackled, which is restoring RBM20 mutations in vivo by gene editing, which this approach in principle could be agnostic of mechanism. It doesn't need to know mechanism in order to target these variants. The idea here would be directly correct the alleles if we can get an editor delivered to the cardiomyocyte system. Several challenges associated with genome editing agents and applications, in particular when applied to in vivo contexts. So one is to achieve tissue or cell type specificity of the delivery, mainly as a way to overcome any toxicity of off-target effects or immune responses, because if you can target the delivery more precisely to the heart, you need to use less dose of your virus and also of your editor as you achieve more specific effects there. We primarily resorted to viral delivery systems based on AAV vectors and adenoviral vectors. These are small, non-pathogenic viruses. They're engineered to deliver genome editing agents. They pose a relatively low immune risk, and they can affect, which is important for the heart, both dividing and non-dividing cells, since cardiomyocytes are post-mitotic and no longer dividing, and potentially offer longer-term expression of our delivery constructs. What was, for us, key in this application was that we used a virus that was modified and optimized to target heart cells over the standard AAV9, which primarily goes into the liver. And so you need, for standard AAV9, you would need to use a lot higher doses. But because we had a capsid variant that's called AAV-Myo developed by Steinmetz Lab, it primarily went into the heart, and you can see here that it goes primarily into the heart and very low site or off-target delivery to the liver, which is primarily the case for an AAV9. In terms of editing constructs, we have several possibilities of editors that we could use to apply in the context of the heart to achieve gene repair. Unfortunately, homology-directed repair-based systems have very low efficiency in the heart. They rely on an NHGRI mediated mechanism that is active during DNA repair processes in cell division. And so we don't have cell division occurring in the heart. So we need editors that work there. And for that, luckily, there are base editors that are available. So we can use cytidine or adenine deaminase or prime editing, which also doesn't rely on HDR mediated repair. We decided for base editors, primarily because the efficiency is higher for base editing than for prime editing. And lucky for us, both the mutations we wanted to target, the P633L mutant and the R634Q mutation, can both be targeted with an adenine deaminase. And so we could use the same editor for both of these two mutations. You would get a deamination and a transition mutation that occurs here in the absence of a double-strand break. which has also been described as a beneficial effect for mitigating unwanted secondary mutations at the target site, so-called bystander edits. We had to package these editors into the viral constructs. Unfortunately, the packaging capacity of the AV wasn't sufficient to put all of it into a single AV, so we had to use a strategy where we split the Cas9 protein into two vectors, but we use a split intine strategy that would then bring these back together, and we're ejecting about 10 to the 12 vector genomes per mice into these animals, again doing an injection at 4 weeks of age, and then we'd analyze them later, mostly at 16 weeks of age. This shows you the editing efficiencies that we achieved. So we used the adenine deaminase packaged in the AV myovector and compared that to what would happen if we had just used AV9 in terms of editing. And you can also see here, similar to the data I showed you earlier, that AV myo achieves a higher editing performance. And this is DNA analyzed across the entire heart, which obviously consists not only of cardiomyocytes, but also other cell populations that are not targeted by the vector. So we have about a 15% edit efficiency. If we look across the whole DNA of the heart, that doesn't measure the target efficiency yet of our editing system. I'll come to that in a second. It was a lot more specific, again, for the heart than we got editing in other tissues of the body. Very low editing, for example, in the liver, which is consistent with this vector data that I showed you earlier. And to our pleasant observation was that we get fairly efficient edit performance. In fact, 70% of the alleles of RBM20 were repaired in the heart in vivo with these editing combinations. So the difference between editing measured at the mRNA level versus the DNA levels, that RBM20 is only expressed in the cardiomyocyte population. And so by measuring RNA editing of the RBM20 alleles that are present as mRNAs, we can directly measure the efficiency of our editing constructs, which are delivered to cardiomyocytes specifically. So it's 70% edit efficiency was good, and we obviously then were curious to see if the phenotypes of RBM20 DCM are also restored in this system. So we analyzed splicing defects, and they're repaired. Localization is also restored. So RBM20 now, if it's not mutant, it doesn't stay in the cytoplasm, but gets correctly localized to the nucleus. And again, pump function as the most important readout in these animals. Pump function is completely repaired, and we see wild-type pump function in animals where gene correction has occurred compared to the mutant animals in the absence of repair. So questions, of course, that come up is how specific is this and how many potential off-target effects do you see? So we carried out various genomic characterizations of these animals, including single nuclei RNA sequencing, where we isolate the hearts from mice that have been edited and then sequence about 40,000 nuclei in this experiment from 3 different mice that have been edited, and analyze the populations of these cells. We get all major 11 cell types of the heart are showing up as transcriptome data that we get out, including ventricular cardiomyocytes and atrial cardiomyocytes. And of course, the key question is, where is the editor expressed in these cells? And as we would expect with an AV myovector that primarily infects myocytes, we also get base editor expression specifically in the myocyte population of the heart, the ventricular, the atrial cardiomyocytes. We get no expression essentially in the other cell types of the heart, which is consistent with specific delivery. And we were wondering if the gene expression profile then of edited cells is also restored back to wild type levels. And so for that, we looked at the whole transcriptome, both for RBM20 target genes, but also globally in ventricular cardiomyocyte populations. You can see here the RBM20 mutant animal derived cells, the wild type, and here the edited in the middle have an intermediate transcriptional profile, which suggests the profile has reverted back to a more wild type state in the presence of editing. What about other cell types and their transcriptional state? The editor is specifically changing the alleles in the cardiomyocyte populations, but neighboring cell types like fibroblasts, epithelial cells, parasites, myeloid cells also appear to be transcriptionally corrected in these hearts. Not surprising, right? Because your heart functions as an entire unit. If you fix the disease cause and origin, it will have a beneficial effect on the entire heart function, including non-edited cell populations that are part of the system. What about off-target effects? So here, to quantify that, we use whole genome sequencing of these mice hearts and compare the allelic profiles in the heart versus other tissues in the mouse. So in this case, liver and tail tissue was collected and also sequenced. The vector, as I mentioned, is going specifically in heart. We have no delivery to these other systems. If there were substantial off-target edits that occurred, we'd expect a higher rate of mutations in the heart compared to these other tissues. We don't have any evidence of that. You can see that here, even for the nature of the substitutions that would be mediated by an adenine deaminase. We don't see elevated rates of mutations in the heart versus other tissues. That suggests to us there's no large-scale aberrant off-target mutations that are being introduced by these editors. This is still, I should tell you, bulk sequence data. So if there are individual cells that have different mutations that happen at random and low frequency, the whole genome sequencing wouldn't pick it up. But there's no systemic off-target mutations that are being introduced by the system. And in fact, you can also look at whether any of these variants are near guide RNA similar sequences. So if the guide RNA would have directed the enzyme Cas9 editor to that location, you would expect edits near guide similar sequences in the genome, but they're not sequence related. So together, the editing suggests that we got 70% allele correction in the heart, in the cardiomyocyte population. That was sufficient to restore the ejection fraction in these animals. We injected animals that are juvenile, so 4 weeks of age. At this stage, you have molecular malfunctions in terms of RBM20, but you don't have any significant ejection fraction decline yet. The key question for us now as the next experiment is to think about animals that are a little bit older and to see whether we can correct the disease and have a beneficial effect if we go in later. What we could also show is that we see transcriptome correction beyond the cardiomyocyte population affecting all major cell types of the heart, and we see no major systemic effects of our off-target edits. In principle, a gene correction strategy would be applicable to all forms of familiar dilated cardiomyopathy, as long as you can get an editor that functions within that region and you have a PAM sequence close by that you can target, or you can get your editor to target the mutational status. So potentially this type of strategy, which doesn't rely on a mechanistic understanding of disease, but requires identification of the causal variants at the genetic level, could be applicable to a large set of patients and diverse types of disease genes. So what are next steps? I want to spend the last just few minutes on talking about what we're additionally working on within this area that's ongoing. I mentioned already we'd like to go into animals of later ages to address this question of whether we can not only prevent the disease, but whether we can also cure it in individuals that have developed the disease. So for that, we're taking mice with the RBM20 mutations at older ages, 16 weeks and 1 year old. We would analyze whether editing can restore heart failure after disease onset. We would also, again, analyze these animals for off-target edits and also look at how the tissue microenvironment is influenced by our editing actions using spatial transcriptomic methods. And so this is a project that's not just now getting started. Second angle that we've taken, where we've done a little bit more work on, is to take this concept of trying to stratify patients into treatment classes. Now, based on our insights of what happens in this disease, and we think we can come up with tailored therapeutic recommendations depending on the type of mutation, even within the RBM20 gene that patients see. So I come back to this picture, which actually shows you the RS domain mutations shown here. I showed you these mainly lead to mislocalization and thereby missplicing. There are also mutations in other domains, including the RRM region, which is involved in RNA recognition, and the E-rich region. And the approach we wanted to take here is systematically change every single amino acid to every other possible amino acid throughout these regions to do a saturation editing experiment that we can then use our genomic tools, and you'll guess it, image-enabled cell sorting, to then systematically determine which mutations lead, for example, to cytoplasmic localization. So these experiments can be done at high throughput. We simply design oligo libraries that encode every single variant possible within these domains. So I show it to you here for the RS domains, a 60 amino acid window. We change every one of those to any other of the 19 amino acid possibilities at that position. We clone that entire library into lentiviral plasmids that we can then transfect into either HeLa cells or iPS-derived cardiomyocyte systems, and then sort them on the image sorter to sort out the populations where RBM20 is nuclear versus RBM20 is now cytoplasmic due to the mutation. And we simply then sequence our pools of RBM20 genes by sequencing this entire domain and determining which mutants lead to cytoplasmic localization among these amino acid substitutions. And this has been very interesting to do that for, we've now done it for the RS domain and the E-rich region. And what we really see when we do the entire library for the RS domain is we get very specific hits that are mislocalizing RBM20, and they're exactly clustered in this hotspot region that I showed you before, where the 6 amino acids in a row have all been leading to this disease. And exactly those mutations are also the ones that lead to mislocalization of RBM20. But remember that in each of those families, there was one amino acid substitution. This data suggests that essentially most amino acid substitutions within those positions are going to disrupt the localization of RBM20. So presumably patients that come around that might be still detected in the future could, we would think, have mutations of similar types that so far haven't been detected, but lead to RBM20 mislocalization within that region. The E-rich region isn't involved in direct interaction with TMP03, and so substitutions within the E-rich region are not leading to cytoplasmic localization. They still lead to an RBM20 DCM, presumably because missplicing occurs in these mutations without the RNP granule formation. And so we're now complementing these types of libraries with a detailed transcriptional readout too, to intersect how transcriptional missplicing and mislocalization coincide. But with that, we hope to generate kind of a lookup table that could then be applied to patients' data more broadly. So ultimately, we think we can stratify patients based on mutations into our 3 treatment strategies. So RBM20 mutations, that have where you see no granules, but you might have low RBM20 expression and low splicing, for example, for a heterozygous nonsense mutation or an E-rich region domain. Overexpressing RBM20 or activating RBM20 expression would be therapeutically interesting for these individuals because it would overcome the haploinsufficiency on splicing that's seen. For the RS domain mutations where mislocalization is seen, but they're actually, the variants are splice active. If they get to the right place, you could relocalize RBM20 back into the nucleus and you would fully restore splice activity. And then of course, any variant could benefit from gene repair once this strategy works in directly fixing the alleles in the heart and correcting them before diseases set in. So finally, last couple of slides, and I'll go very quick here. We also in the lab think about ways we can detect this disease earlier in order to prevent it from arising and not rely on echocardiograms or ECGs and patients going to the hospital to detect these diseases. And here we're thinking about wearable devices that could be applied in these contexts. For IBM20, we don't have anything yet, so I'll show you some examples of how we think about this within the lab. And you can divide sensors into different categories, your portable, wearable sensors, edible sensors even, or implantable sensors. The glucose sensor, for example, for diabetics is an example of an implantable sensor that could measure biochemical properties of the system. We're hoping to develop sensors that can rely on easily accessible bodily fluids in order to do analysis on those. One example of a technology that my lab had worked on about 10 years ago, developed or used magnetic levitation technologies in order to, as a new way to physically separate biological molecules and cellular systems. You can see an experiment of the frog here on the left side, carried out by NHGRI, who in the year 2000 got the Ig Nobel Prize for this experiment. He levitated a live frog in a high magnetic field, 16 Tesla or so magnetic field. It stayed alive after that. We were interested in applying this concept to individual cells. So could we levitate individual cells? And so we, together with Utken Demirci's lab, developed one where we can levitate cells in a high, about 3 Tesla magnetic field by bringing 2 stationary magnets very close to one another that can generate a large density gradient of magnetic field strength there, sufficient to levitate single cells. You can see cancer cells that are clonal levitating here at the height that corresponds to the cell density. You can register real-time changes in this through stimulation. In this case, if you kill the cells, they change their levitation height. So you can use this both as a way to sort out cellular populations or to measure perturbations on them. So for example, applied here to isolate circulating tumor cells out of blood. Tumor cells have a different density than red or white blood cells have, so they levitate at a different height. If you flow them through this magnetic chamber and then collect the top and the bottom fraction, you can collect blood cells in one and tumor cells in the other and characterize that more. That has also led to a spin-out company from the lab. So this device is now commercially available by Levitas in order to separate cells and has potential applications in the cardiomyopathy space in cases where these mutations lead to different lipid fractions in the cardiomyocyte population. I think I'll finish with this last slide. One of the sensors that we think we can adopt to more molecular readouts that ultimately could be interesting in the context of DCM2 is a technology where we generate DNA nanoballs based on amplified nucleic acid, and we detect them through electrical impedance detection, where a DNA nanoball, negatively charged due to the DNA amplification and also the size that it has, would perturb the electric current across 2 electrodes. And so this simply was inspired during the COVID pandemic, where LAMP reactions were being utilized to amplify SARS-CoV-2 virus. We could amplify it large enough to generate a DNA nanoball that's simply formed by compacting that amplified nucleic acid using compaction oligonucleotides to pack it together. These can generate balls of about 1 micron in size, sufficiently large to see under the microscope and also sufficiently large to detect by an impedance change as they're flowing through 2 electrodes. So potentially sensors like this could be developed for molecular monitoring technologies on portable devices. So with that, I want to finish. I told you a lot of approaches to RBM20 DCM, inspired by genetic analysis to start with, to characterize these mutations, develop model systems, iPS cardiomyocytes and animal models, characterize the splice patterns of these mutations, think about more systematic variant effect catalogs for RBM20 genes to then classify patients into different therapeutic strategies. And in terms of our strategies, understanding RBM20 mislocalization was key, but also gene therapy applications we're very excited on and taking those further ultimately into a clinical setting. Our next step is pigs, and after that, human systems. And finally, I didn't talk a lot about it, but early detection of biomarkers using some advanced sensing technologies are still a little bit out there, but we're hoping to develop a little bit more on that space. So with that, I want to finish. Let me skip the last 2 slides and just acknowledge everybody who contributed to this work. Sorry, both in my lab at Stanford and also in Heidelberg, and many collaborators that have contributed. So thank you.
Thank you for the really comprehensive overview. I'm curious, have you done any like reverse genomic analysis to look at RBM20 to see, like in All of Us data set or UK Biobank, like what is the mutational spectrum? Yeah, right. Are there people walking around that have a pathogenic allele and have no evidence of cardiomyopathy? Or then again, are there lots of younger people that are taking, heart failure medicines and no one knows why?
No, I think it's so we did look a bit in the NOMAD database. for, variants that are found there. There are actually, we were surprised, there are quite a number of nonsense mutations within RBM20 that are found. If you overall look at the frequency of these mutants, it's less than what you would expect based on the size of the gene, right? And then, random nature of mutations during DNA replication that happen, right? You can calculate how many you might expect. You see a depletion of those, which suggests to me that there is an important role of RBM20 during development. Even if you have a heterozygous nonsense mutation, you're likely not to make it to life. But if you do survive some of the development, some of these individuals make it through. The question there, of course, is, is that data from still young individuals that, are carrying these variants that, and they're probably less severe than the RS domain mutants, has the disease simply not developed yet, right? I don't have any data on the age classes and so forth. I have not looked at that.
And from your clinical information on the patients, if you notice, depending on your mutation, at the end you were showing how different domains have different mechanisms. Do those patients need different pharmacotherapeutics? I mean, would you change your medicine regimen in such a patient if you knew their mutation?
Which mutation they have, So I think that's, I would say, based on the insights we have on localization of RBM20, particularly the mislocalization, I would suggest patients that have a mislocalized RBM20 will get more aggressive therapy applied to them than patients that only have the splicing defect, because they're lacking the dominant negative effect. And we don't have yet a detailed view on the mechanism that how the granules lead to the disease. One of our favorite hypotheses is that they sequester mRNAs that are important in other processes in the cell, including energy metabolism, as you mentioned earlier. So, but certainly with those ones, you wanted to pay more attention also to arrhythmia, because one of the concerns with this disease is the sudden cardiac death that patients see due to a ventricular tachycardia event. So, probably, defibrillator implant would be recommended at at least in an earlier stage, at least for the patients to be aware of it.
Great. I'll let someone else ask a question. Thank you.
Yeah.
Sorry, talking about base editing, you said DNA level efficiency was around 15-20%, but RNA level is like 70%.
Yeah.
So is this something unique to this gene or this disease, or this is happening all the time?
Right. So let me explain that. Yeah, because that's sort of so the DNA measurement came from extracting DNA out of the whole heart, which obviously has cardiomyocytes, fibroblasts, other cell types in the heart. But the vector, the AAV vector we're using is targeting specifically cardiomyocyte population. So a DNA level analysis underestimates the efficiency of editing, because with that, we're getting DNA in there from cell types where editing couldn't even have occurred. And so the RNA editing gives us a more direct readout of just specific RBM20 editing in cells where the vector has reached them. And that's the cardiomyocyte population that uniquely expresses the RBM20 gene. So the 70% correction is what we see on the RBM20 allele when the cell has got the editor constructs delivered to them.
So RNA, it was not isolated from the whole heart. It's only
correct. No, the RNA is isolated from the whole heart, but only the cardiomyocytes are expressing RBM20. It's like a cardiomyocyte-specific expressed gene.
I see what you're saying.
Yeah.
Okay.
I have a question about the iPS-induced cardiomyocyte differentiation, because I did this similar experiment before, and I realized that the cardiomyocyte splitting is kind of like random. It's, if it's immature stage, it's spontaneous. So I'm just wondering, like, when you just try to say, okay, this mutated cell line, they have some arrhythmia or they have a slower beating. So I'm assuming you probably guide the cell into the mature stage, and just wondering how you
you're touching on an important problem in the iPS cardiomyocyte differentiation. One is that all of these differentiations, no matter how mature your protocol is, are generating embryonic or fetal versions. I think more embryonic versions of cardiomyocyte populations. So you don't reach the adult stages with that. We try to collaborate with the people that have worked on this for many, many years, Mark McCullough's lab in particular, and Joe Wu, to generate populations that are more robust to our end. So we use their maturation media, which supposedly differentiates it a little bit better. But it's something to keep in mind in these systems. I think for cases where you use that to study molecular properties of the cardiomyocytes, like transcriptional states, splicing activity, or mislocalization, the differentiation is sufficient enough to uncover those phenotypes. But I wouldn't rely on measurements at the physiological level that might reflect more what you'd hope to see in an adult system, because you're dealing with an embryonic version of that. Yeah.
Thank you.
They're very powerful for all kinds of genetic screens, right? That's where the cell systems really help us, besides the in vivo models. But the phenotypes are ultimately robustly detected in the in vivo.
You showed a whole bunch of variants at the beginning. I was wondering if you would kind of investigated any of those too, and
yeah,
how that's all going.
Yeah, the other ones. Yeah. So the only place we have them integrated is in our systematic library, where we actually did all possible amino acid substitutions. So that also includes the known mutations within those domains that we targeted. So we haven't done any animal models yet for other RBM20 mutations. But one idea to follow some of that up, would be to take interesting hits that we get from the variant effect mapping and put them into an animal system and study those more. Yeah, so good ideas.
And I guess related, all the genes you showed at the beginning too. Have you guys investigated?
Oh, the other genes, yeah. We've been really laser focused on RBM20 to try to push this forward, because we ultimately want to reach a clinical state to bring it in, and we have plenty to do there to get that to go. So we have not actively worked on any of the other ones. We collaborators of ours sometimes have picked up some, and in some cases they have an interaction effect with RBM20, and so we're involved in some collaborations there. But yeah,
thank you.
Yeah.
All right, if there are no other questions, thank you, Lars.
Thank you very much for staying late. Thanks. Yeah.
Since you have that mutation cluster,